Okay, so we're going to start with a review of DNA. We're going to go through, so we're going to use the foam kit for this one. So you can see these little foam pieces. They each have different numbers, letters on them. So these foam kit pieces are going to be our nucleotides. Okay, nucleotides, remember, are a macromolecule. It is our nucleic acid monomer. Okay, so in our nucleotides, we have three parts. We have a ribose sugar. Okay, we have a phosphate group, and then we have a nitrogenous base. Okay, there we go. Now our nitrogen bases are going to be the part that differs. So for each nitrogen base, we can have an A, a T, a C, or a G for DNA. A was always going to bind with T, and C will always bind with G. So if you remember, that is Chargaff's rule. Okay, A will always bind with T, C will always bind with G. So we're going to set up our DNA. So if we look at our nucleotide right here, our phosphate group is going to be that part right there that goes into that. And this part right here that sticks out, that arrow part, is our hydroxyl group. Okay. All of these are going to bind together. So they're going to come together in a long strand. Okay. And it doesn't matter for our activity today what order they're in. But you can see that we have these nucleotides that are all going to be lined up. Okay. Now, remember, DNA is double stranded, so we have the strand on the bottom, and then we're going to have the strand on the top. Okay. Now, we said A always binds with T and C always binds with G. So above each A, you're going to put a T. Above each G, you're going to put a C. And if you look, your A's will only, see how that has a curve, will only fit with our T's. Okay? And they're going to click together. So we're going to have our mini DNA strand here. All click together. Double stranded. It's going to look something like this. Okay, so you have A's matched with T's on top. Okay, T matched with A, a C matched with a G down here matched with a C up there. Okay, now you notice that those arrows are all pointing this way, whereas these arrows are all pointing this way. They're pointing opposite ways. Okay, remember that. They're going in opposite directions. This strand is pointing that way, and that strand is pointing this way. Your DNA, this right here, our sample DNA, is inside every cell. Where in the cell is your DNA located? It's going to be in your nucleus. Now, every cell in your body, every somatic cell, every body cell is going to have the same DNA in them. Remember when we talked about cells, do all of your cells look the same? No. Remember how we looked at nerve cells, and we looked at muscle cells, we looked at skin cells, we looked at sperm and eggs. They all look different, okay? But they all had the same DNA in them. Okay? And then you talked about cell division. We talked about mitosis and meiosis and how this mitosis DNA is replicated so that it can go through mitosis and you end up with more cells. Cell division for as you grow and as you heal. Okay, so we're going to take our DNA strand that we have here and we're going to simulate DNA replication. Now with this kit, we're going to have a little longer DNA here. We're going to have a whole strand of DNA, which we then get to replicate. Okay, so if you're following along in the packet, you should be about page 4. So on page 4, it's showing you that one strand goes in a 3 to 5 direction, 3' to 5'. The other strand goes from 5' to 3'. Okay, this is called anti-parallel strands. Okay, what you need to remember is that your hydroxyl group is always going to pair with the next phosphate. Phosphate group inside hydroxyl group point, matching with that phosphate group, matching with that hydroxyl group. Same thing on that side, they're just pointing in different directions. So when we get to our kit, we have this, okay, now it's going to say DNA replication at the top, we're going to have the orange one that has a little bit there so you can see. This orange part is your helicase. Remember, anything that ends with an ASE at the end is going to be an enzyme. Okay, so our DNA is going to come into this enzyme. Orange is the enzyme, your helicase. We're going to go through a couple different enzymes. You need to know this one. Okay, now as it is being pushed into there, what is happening here? See, the form of the enzyme right here is going to be splitting that DNA, and then it's going to go through the other side. So we're replicating DNA, making twice as much of it as what we started with. So as it goes through, it's going to come out this side. So it's starting double-stranded here. It's going to come out this side as being single-stranded. Okay, so your helicase, this orange enzyme, your helicase is going to split open your DNA. And then you have another enzyme. This enzyme, this gray one, is a DNA polymerase. See how it ends in the ASE, meaning it's an enzyme. Your polymerase only runs in one direction. See the arrow? And it's going to show you how it's going to run. Okay, so you have an active site here. Remember, the active site is where something happens. And it is going to jump on each time you go through replication, you're going to have 2 DNA polymerases. One of them is going to start here, and it has that active site. And as it goes through your active site, so this is going to push through. We have our active site right here. Your DNA is going to come up to that active site right here. Okay, and you're going to grab a matching complementary nucleotide to attach to that. And then it's going to move through the next one. Okay, so we have a polymerase doing the top strand, and then we're going to have a polymerase doing this bottom strand. So as the strand comes through, it's going to be matching with that complementary base pair. So every C gets a G, every T gets an A, every A gets a T. And we talked about how this DNA polymerase, ACE meaning an enzyme, only functions in one direction. So as this comes through, it's going to go through this enzyme. This enzyme is going to keep making the complementary strand. Just as fast as it comes through, this one is going to keep binding. Okay, this is going to be your leading strand. Okay, because it can synthesize continuously. Okay, as long as it's being pushed out of the helicase, it's going to be going through there. Your DNA polymerase will keep matching it on this one. Now on this one up here, this is your lagging strand. Okay, so what's going to happen is as this comes through, your DNA polymerase has to bind It's going to bind here partway through the strand, and then you're going to bind there, and then it's going to move a little bit. You'll have that complementary base pair. It's going to move a little bit more. You'll have that complementary base pair. Move a little more. You're going to add that one in. Okay, this is called your lagging strand, because then your DNA polymerase has to come off, wait for more DNA to go through, and then jump back on again. And this is going to form little fragments of double-stranded DNA. These little fragments are called Okazaki fragments. Okay? Okazaki fragments are these little strands that form in your lagging strand. Leading strand forms continuously, lagging strand forms discontinuously. Okay? I want you to go through, watch this again, watch the other videos. We're going to go through a little more replication, and then transcription and translation. Replication is taking one strand of DNA and making it into two. It's a semi-conservative method because this is your original strand here with this original strand down here, and each one of your original strands is also part of your product. So this is a new strand bound with an original strand, a new strand bound with an original strand. Semi-conservative. So you always have that mold, okay, that key that's working the new part. Okay, it's part of a check and balance system. So go ahead and walk through the rest of this, see what questions you can answer. Watch the videos in the playlist, okay, through that Central Dogma playlist that I have on YouTube. Okay, and then come back and we'll be ready to do the transcription video. Okay.